Hi everybody, welcome back to class again. Today is Thursday, which means that we are going to have our biology lesson. Usually on Thursdays, we're going to try to do a biology lesson every week, as we said before. So today we're going to start off by looking at carbon and organic macromolecules. This is looking at the pretest. This was one of the weaker areas. So to begin, we're going to look at carbon. And carbon is the backbone of all organic macromolecules, but also all organic molecules. When you study organic compounds or organic chemistry, you are studying carbon-based compounds. So carbon is the most important atom here. And carbon has 4 valence electrons. And what we mean by valence electrons are these dots here on this model of carbon off to the side here. Those are valence electrons. That's how many bonds carbon can form. And because it has 4 free valence electrons there, it can also form 4 covalent bonds, which is very important, which is why carbon makes the backbone, is because of the way it can bond and the number of bonds that it can form. And we can see pictures of it here where you can have carbon form 4 single bonds. You can have carbon form 2 single bonds and 1 double bond. This bond right here, that would be a double bond. And then you have over here where carbon can form a triple bond and a single bond. In all the combinations though, each one of these lines makes a bond, and you can see 4 of them in each one of the combinations. Now, whenever you are looking at carbon, or organic molecules, let's say, and you see them on the keystone or you see them in other places, a lot of times you're going to see them as structural formulas. And a lot of times in structural formulas, you're going to see them drawn where the carbon and hydrogen atoms aren't drawn and they're just there, they're understood, meaning like this, where you see that line structure. You don't see anything there other than these little bends. But at each one of these bends, like right here, that little point there, that little point is actually right there. It's showing you that there should be 2 hydrogens and a carbon there. But anytime you see that little bend there, right there, that point where it bends off, that's a carbon atom. So then this point here would actually be right there. Here is here. And then actually down here where it ends, instead of having 2 hydrogens, you'll see 3 hydrogens attached to a carbon. Those are called the line structures. And they're just lines that have bends in them showing where the carbons are. And that's how you'll usually see them. And we're going to look at carbon a little more here. And basically what I'm showing you here with these structures is to transition from these structures over to line structures and how you might see them and how to recognize hydrogens and carbons, knowing that right here is actually right there. Here is here. That's the point of the line structure. It's shorthand. It's a lot easier to draw the structure that's above like that than draw all the carbon, 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 hydrogen. It's a lot faster to draw those. So what we have over here with these line structures off to the right is just right here off to the right is just going through practicing drawing them and recognizing how they would be drawn. So in this first structure here, this is actually a benzene. That's a benzene ring. And when you're drawing this, you're going to, everywhere there's a carbon, there's going to be a bend. So you're going to see, I can't draw a hexagon very well, but that would be your benzene. And then you would add where you see this double bond, you just add another line, another line, another line. And that's your benzene. That's all there is to it there. Right here you have T, butanol. You have T-butanol. So to draw this, it's real simple. It's going to look like a cross with an OH. Anything else other than carbon and hydrogen will get drawn. So this OH will get drawn. It just doesn't have all the pieces like that does. And then the last one here, when you're drawing chains, this here is called a chain. This would be a ring. When you're drawing chains, you'll start from the beginning and then go up, whoop, down, up. So we have 1, 2, 3, 1, 2, 3. And then at the end here, you would draw your NH2. NH2. Nitrogen, nitrogen with the 2 hydrogens, NH2. And we have 3 carbons, and we have 1, 2, 3 carbons. This is complicated. It will get easier as you practice it and as you mess around with it. So moving on from carbon, what we're going to see are things called functional groups. Now, these functional groups are going to help you identify organic macromolecules. And that's always a question on the Keystone test is, what is this image? What type of organic macromolecule is this? Is it a protein? Is it a carbohydrate? Is it a lipid? Or is it a nucleic acid? And these groups will help you identify it. When you're looking at the functional groups here, these are the only 3 that I'm going to be saying that you have to remember. You have an alcohol group, or another name for it, hydroxyl group. You have a hydroxyl group. And all the alcohol or hydroxyl functional group is an oxygen bonded to a hydrogen at the end. And we usually don't draw that little bond in here. It's just like this, OH. So anytime you see an OH by itself attached to a carbon here, it's attached to this carbon, then it's an alcohol or a hydroxyl group. The next one's going to be an amine. It's called an amine. An amine is normally NH2 is how you'll see it. It's going to be NH2. Sometimes like here, it looks a little different because it actually has a carbon bonded to it right here. It's still an amine even though it's only 1 hydrogen though. But anytime you see, like even in that previous example, here, example right here is what I was talking about. That would be an amine group. Anytime you see that NH2 just sitting there, that's an amine. You'll see a lot of amines with your proteins. Then the last group is a carboxylic acid. A carboxylic acid. This one, it looks like an alcohol, but it's not. It has that OH group here, but it has more attached to it. It has an OH with a double bonded O. Those 2 lines like this are double bonds. So you have an OH with a double bonded O. OH with a double bonded O. They're only going to show up at the end of molecules, either on the far left or the far right end, because of how they're put together. This functional group is a kind of bulky group, so nothing more can bond to it. This carbon here in the middle, it ran out of bonds. It has 1, 2, 3, 4. So it always has to be on the end. But that's going to be your carboxylic acid. You're looking at it for that double bonded O, double bonded oxygen, bonded to that OH there. You're also going to see this one here with nucleic acids and your proteins. Now you'll see a lot of times this R down here. You'll see this R attached to groups, and anytime you see R, it's just generic. It's simple. So for example, if I would take this structure right here, and I didn't want to include all that, the benzene looking ring, I could have just done ROH. This R just means something generic. It's something with carbon bonded to that oxygen there. And as we start to get into the molecules now we're looking at the monomers versus polymers. Monomers are basic units, and polymers are chains of units. So when you see monomer, we're talking one. Monomer means one. Polymer means many, poly. And as you can see, this monomer here of sugar, there's only one ring here. But starch, it's got 1, 2, 3, 4, 5. And these dots that you see right here and here mean that there's more bonded to it. So you see 5 there now, but that chain keeps on stretching out. So that's the difference between a monomer and polymer. So then we get into the actual organic macromolecules. There are 4 of them: carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates, lipids, proteins, nucleic acids. And each one of them have different atoms that you will always see there. When you see certain atoms and you only see certain atoms, then you can almost immediately identify what something is. For example, if we look up here and where this here, organic building blocks, we see carbohydrates and lipids. Oh, I don't like that color. It doesn't show up. Organic building blocks, carbohydrates and lipids are always going to have oxygen, carbon, and hydrogen. Both of them always are going to have oxygen, carbon, and hydrogen. When we add the element nitrogen, now we see amino acids and proteins. But I would like to take it a step further and say we're going to include sulfur in this. When you see sulfur and you see nitrogen, oxygen, carbon, hydrogen, that's immediately going to tell you protein. Anytime you see sulfur in there with it, it's going to be a protein. If you see nitrogen, it's most likely going to be a protein, because it can't be a carbohydrate or lipid, because carbohydrates and lipids only have oxygen, carbon, and hydrogen. And then the last one is going to be, if you see that P, and there's going to be a phosphate group with it, as you'll see later on down the line, it's going to be a nucleic acid. Only nucleic acids are going to have that phosphorus group in there. So anytime you see the element phosphorus or that P, it's an automatic nucleic acid. Nitrogen and sulfur, it's probably going to almost automatically give it away as an amino acid or a protein if you see that sulfur in there. If you only see oxygen, carbon, hydrogen, it's either a carbohydrate or lipid. And we're going to start with carbohydrates here. So carbohydrates are also known as your sugars. They're also known as your sugars. And they have a ratio of 1 to 2 to 1 of carbon to hydrogen to oxygen. So that, no matter what, you're always going to have that simple ratio. Even if you have, even if you have multiple atoms, it's still going to boil down to that 1 to 2 to 1 ratio. In carbohydrates, the monomers that make them up are called monosaccharides. Those are your simple sugars, your one little things. And they can be either in rings, which are these, or chains, which are these. This here again is a ring. Chains are real long lines of them. Your rings look like rings. It doesn't matter what shape they are. This one here is a hexagon. This one here is a pentagon shape looking. It doesn't matter what the shape is, but they're both in rings. Now you will have chains form into rings. Chains will turn into rings because of how they bend around and bond together. Now, carbohydrates could be made of 2 monomers. They could be made of 2 simple sugars only. And when you have them made of only two simple sugars, they're going to be called disaccharides. Di means two. Di means two. Mono means one. So disaccharides would mean two. An example would be sucrose and lactose, where you have the two monomers bond together, and they're going to bond through dehydration synthesis. And what that is a bond formed by losing water. What we mean by losing water is we're looking right here at these OHs. When they bond, you're losing one hydrogen, and one of those OH groups, it doesn't matter which one, but one of them are disappearing. And they bond together to form water here, and a bond forms at that oxygen. That would be dehydration synthesis. Anytime you see water leaving, you're dehydrating. It's the same way that if you lose too much water, you're becoming dehydrated. Continuation of carbohydrates here. So we have monosaccharides as one, disaccharides is two. Poly, we already said, means many. So polysaccharides are very complex sugars. We already saw starch in the one example before, but you also have cellulose, which is in plants, and glycogen. These are very long chains. These are very long chains made of rings. As you can see, this is a chain here, but we have a ring, 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 ring. All of those rings bond together to form these long chains. And that's the basic parts of carbohydrates. The biggest things here out of this section will be the ratio, carbon, hydrogen, oxygen. Carbohydrates are only made of carbon, hydrogen, oxygen. What a monosaccharide is, what a ring and chain looks like, what a disaccharide is, and how carbohydrates bond through dehydration synthesis. And then what a polysaccharide is. And be able to try to identify some of them. For example, some of the common ones like starch or cellulose or glycogen. Be able to identify those as polysaccharides. The last part here is just looking at uses for carbohydrates, and remember these uses. Carbohydrates are used for quick energy. Glucose, sucrose, fructose. Think candy bars. They're simple sugars that give you real quick energy. Some of your energy drinks, teas, anything that has a lot of sugar for very quick energy. You just need something for like that pick-me-up type thing. They can be used for energy storage, like we have the starch, glycogen, and something called pectin. What we're saying by energy storage is these long chains like this aren't broken down very easily, so you can get energy built up with them, and they're long-term energy. They allow it to burn longer and allow you to feel more energized for longer periods of time compared to the candy bars. An example you could think of it would be the difference between putting a whole bunch of newspaper in a fire compared to putting something like a piece of oak wood in the fire. The oak wood is going to burn still, but it burns longer than that little bit of newspaper. The newspaper just burns up real fast and disappears. The newspaper would be your quick energy, your simple sugars. And your oak wood would be your energy storage or your complex sugars. And the last thing is structure, would be cellulose. And with cellulose, you're going to find that in plants. And it's going to help give it that rigidity, that structure. You're going to find it in the cells. So every time you take into a bite of celery and you hear that crunch, what you're actually crunching through is the cell wall and the cellulose that gives the plant its strength. Plants don't have skeletons. They use that cellulose to hold themselves up. For today, that is where we're going to stop with our lesson. We'll continue on with organic macromolecules next week for our bio lesson. If you have any questions about this, please feel free to contact me. But you will be tested on this now. Now that I'm starting to teach this to you, I can test this anytime. Just please be aware of that. Please do study for that. I hope you have a wonderful day, everybody.